Hi, I'm Wannes, and I would like to present to you our study published in the Journal of Molecular Oral Microbiology about how glycerol strengthens the probiotic effects of Limosilactobacillus reuteri. Since we know the oral microbiome plays both a role in disease and in health, we're interested in seeing if we can change this disease state back to a healthy state through the use of pre- and probiotics. As you may know, prebiotics stimulate already present beneficial microbiota, while probiotics, on the other hand, is more the introduction of these beneficial species. While these concepts are frequently looked at separately, we could actually combine them to have a synergistic effect on both, in which the prebiotic can stimulate the probiotic, and we call this a synbiotic. In our case, we looked at a very commonly used Limosilactobacillus reuteri strain, and something characteristic to many reuteri strains is that they are able to metabolize glycerol to an antimicrobial we call reuterin. Now, while glycerol is very prevalent in the gut, this is not the case for the oral cavity, which just potentially limits the efficacy of these reuteri. From a basic spotting agar, we already saw some interesting effects. While the reuteri on its own didn't have very strong antimicrobial effects, I did notice something very strange. When looking at the condition in which the reuteri had glycerol, I initially thought I forgot to spot the pathogen. Upon closer inspection though, there was something strange about the layout of the plate. Upon closer inspection of the plate, I noticed that there was some inhibition of the pathogen next to the probiotic, but this was actually coming from the synbiotic. The addition of glycerol clearly benefits the antimicrobial effect of the reuteri. Aside from looking at the effects on single species, we also looked at the effect of the probiotic and the synbiotic on multi-species biofilms. In this model, we establish a community of well-known oral bacteria together, after which we use it as a biofilm seed culture to form biofilms with or without a probiotic or a synbiotic. After biofilms are formed, they are rinsed, the cells are recovered and treated with a viability dye to then look at the DNA of living cells specifically. Compared to two controls, we looked at a probiotic condition and two symbiotic conditions. Firstly, we saw that the symbiotic was less prevalent in the biofilms than the probiotic, but it did not translate to the antimicrobial effects. While the probiotic barely affected the periodontal pathobionts, both of the symbiotics did show increasing antimicrobial effects. Similarly, we saw the same effects on the anaerobic commensals, but this effect was not seen for the carcinogenic streptococci. Also for the commensal streptococci, the probiotic seemed to reduce their prevalence more than the symbiotic. We can then also look at the total biofilm, and what we saw is that probiotic can form a large portion of the biofilm, while the symbiotics had a larger effect on the total biofilm and also completely changed the composition of the biofilm. Lastly, as these pathobionts in the biofilm can cause inflammation, we wanted to see if there were some host-modulating effects of the probiotics. To do this, we used the same biofilm model, but flipped the discs and brought them into contact with human oral keratinocytes. After incubating them together, we rinsed them, extracted the RNA, and looked for inflammatory expression. When using naive keratinocytes, we saw that the probiotic-treated biofilm did not reduce inflammation, and the symbiotic only slightly reduced it, but not significantly. On the other hand though, when the keratinocytes were pre-incubated with the probiotic, both the probiotic biofilm, but especially the symbiotic-treated biofilm, showed a lower inflammatory expression. This indicates that not only the killing of the pathobionts in the biofilms was sufficient, but there is also some kind of crosstalk between the human cells and the probiotic, which was also on its own insufficient. To conclude, not only adding the beneficial probiotic might be enough, we might also need to consider the diet of the probiotic. Considering symbiotics instead of probiotics may improve the efficacy of the latter. Thank you for listening.